We are happy to present our work on transcriptional chromatin regulation in healthy and diseased heart. Chromatin is a complex of DNA and proteins whose accessibility changes in response to disease etiology or other stimuli. Therefore, chromatin plays a critical role in regulating biological processes. We collected diseased and non-failing control hearts from different individuals and extracted cells from these hearts. Chromosomes were isolated, and proteins involved in chromatin regulation were captured using a specific antibody. Libraries were prepared using the captured chromatin regions, followed by next-generation sequencing. Chromatin activity can now be compared between diseased and control hearts. Cardiac tissues from human hearts are often snap-frozen and biobanked for research purposes. Their sizes vary, ranging from small needle biopsies to a whole transplanted heart. To preserve the unique composition of cells and molecules in these frozen biopsies, it is crucial to handle them properly. Therefore, in our protocol, we introduce several quality control steps, which are tailored toward small snap-frozen biopsy samples. Using this protocol, we successfully isolated chromatin from small needle biopsies from diseased hearts due to aortic stenosis. We identified altered chromatin regulatory regions in diseased hearts compared to non-failing donor hearts. We also studied the correlation between gene promoter activity and gene expression levels. This data demonstrated a sufficient amount of chromatin could be obtained at a good quality, despite the cardiac samples being small and fibrotic changes being present due to the disease. We studied genes near the altered chromatin regulatory regions in diseased versus control hearts. We found that genes in the proximity of regions with an enhanced chromatin profile in patients were mainly enriched for pathways and biological processes related to fibrosis and platelet activation. In contrast, annotated genes from regions with a suppressed chromatin profile in patients were mainly enriched for pathways and biological processes related to the cell cycle, which stayed in line with the established knowledge in diseased hearts. Using the same protocol, we also obtained chromatin regulation profiles using myectomy samples from hypertrophic cardiomyopathy hearts. Additionally, transcriptomic and proteomic profiles were obtained as well. It can be appreciated that profiling chromatin regions detected many more changes compared to gene and protein levels. More than 12 cell types are present in the heart, and their composition changes with different diseases. We examined the chromatin activity of promoters of cell-specific marker genes in both diseased and control hearts, including cardiomyocytes, fibroblasts, and several immune cells. From the altered chromatin regulatory regions, we also identified transcription factors that were likely to bind to them, and these transcription factors could present as essential regulators of enriched pathways. By integrating altered chromatin regulatory regions with transcriptomic and proteomic changes in diseased versus control hearts, we also obtained a robust set of protein-coding genes that might play an essential role during disease development. For example, HSPA2, a mitochondrial and energy metabolism gene, showed consistently higher levels in diseased hearts from our omics data sets. We confirmed the activated HSPA2 level in the diseased heart by Western blot, immunohistochemistry staining, and immunofluorescence staining. In conclusion, this paper presents step-by-step guidance, including handling and preparing snap-frozen heart biopsy samples from patients and organ donors, extracting enough chromatin at a good quality, and preparing sequencing libraries from captured chromatin regions. After comparing the accessibility and activity of those chromatin regions between patient and non-failing control hearts, we can identify those altered chromatin regions in the diseased hearts, perform many downstream analyses, and integrate them with other omics datasets to screen for robust candidates for the disease. Chromatin immunoprecipitation sequencing, ChIP-seq, protocol for small amounts of frozen biobanked cardiac tissue was published by Methods in Molecular Biology.